Hello, good morning. Myself, I am BSR Botany lecturer. Today our topic, transcription unit, another gene. So what is the meaning of gene? It is the functional unit of inheritance. It is nothing but DNA sequence. Now very important content we are discussing. Look at here. In prokaryotes, the gene is polycystonic. In eukaryotes, the gene is monocystonic. Polycystonic, this is the DNA. You know, the DNA 3 to 5 prime strand forms mRNA. mRNA always present in 5 to 3 direction, formed in 5 to 3 direction. So DNA is forming the mRNA. This mRNA involved in protein synthesis. Especially here in prokaryotes, that mRNA involved in synthesis of many proteins. Many proteins are synthesized because which containing many promoters. Due to the presence of more promoters, more proteins can be synthesized from one mRNA. Whereas in eukaryotes, there the gene is monocystonic, so the DNA forms mRNA, but here this mRNA is synthesizing only one protein because which containing only one promoter. That is important point. Then when you go for another important thing, in prokaryotes, yes, the 3 to 5 prime strand, DNA strand acts as template. That template forms mRNA, that is always forms in 5 to 3 directions. That is okay. That process is called the transcription. Then mRNA involved in protein synthesis, that process is called translation. Both the transcription and the translation takes place within the cell only. There is no compartment like cytoplasm and nucleoplasm. Once the mRNA synthesis takes place, directly which is involved in protein synthesis. Whereas in eukaryotes, just a change. In eukaryotes, this is the DNA strand, 3 to 5 prime strand. It is a template strand. Look at there in the template strand, there which consists of both the exons and the introns sequence. Exons, that is coding sequence, useful nucleotide sequence. Introns, it is not at all involved in coding. It is unuseful nucleotide sequence. So exons, introns, again exons, introns, exons. So the intervening nucleotide sequence present between the exons. So the nucleotide sequence, unuseful nucleotide sequence present between the exons is called intron. Anyway, that DNA forms mRNA by a process called transcription. Even this mRNA also, both the exons and the introns are present. Hence that mRNA, now it is called hnRNA. It is not mRNA, heterogeneous nuclear RNA, which contain both exons and introns. So that is why now the next step is splicing and tailing takes place. Splicing means removal of the introns. Removal of the introns is called splicing. Splicing takes place by spliceosome enzyme. Spliceosome enzyme. So that is why a removal of introns. When the introns are removed, all exons are bind to form a complete mRNA. Now mRNA is forming after removal of introns. Not only splicing, and here two steps are there. Look at here, capping. Capping means, especially in the 5th prime, there guanine triose phosphate can add. Whereas at the third end, adenosine nucleotides, near about 200 to 300 nucleotides added to the third end. Whereas at the 5th end, guanine triose phosphates can add. So like that, complete mRNA forming, that mRNA involved in protein synthesis. So transcription takes place in nucleoplasm, translation takes place in cytoplasm in eukaryotes. So this is the transcription unit and the gene small information. Thank you to all.